Hello, everyone. My name is Mr. Baker. This is the content video for my environmental science class. We're on Unit 5 talking about modern genetics, and this is the second lesson in that unit. Today, we're going to talk about how genetic engineering is used by scientists. And I just want to start off by saying this is in no way exhaustive. It is also kind of dated. I'm discussing this in the beginning of 2021, so if you're watching this later down the road and I haven't updated it yet, then it may be a little out of date. So, the easiest way to think about genetic engineering is that humans have been doing a lot of it for a long period of time, and we're talking about centuries here. So, the easiest way to think about it is selective breeding, and you can think about selective breeding in plants or animals. The easy one for most, at least Americans, is dogs. We have a lot of domestic dogs. They all came from a common ancestor of the gray wolf, but in different areas of the world, they've been bred specifically for certain traits. And so dogs that had those traits were bred together so that their offspring would also have those traits. And they've turned into the large variety of dogs that we know today. And those dogs can still interbreed with one another usually and produce viable offspring. Some of them cannot, but most of the time they can. So they don't have different species, but we have different breeds. The other one is when we think about plants. So plants have been bred for certain traits like larger fruit size or higher yields, or they are better resistant to droughts or to pesticides. And the easiest way for farmers to have done this is to just take the seeds from the plants that they liked the traits of and plant them. Nature kind of does the rest on that. So if you are only planting the seeds from plants that have the specific traits that you want, then the next generation of those plants is going to be more of the plants that have those traits that you want. Right? And you can kind of see that in the diagram where if you breed the two tallest plants, the next generation has a taller population. The kind of newer way to talk about it with modern genetics is when we talk about something called exogenous DNA. So exogenous DNA is DNA that is from another species, right? The kind of fad picture of this was cats that glowed in the dark or under specific types of lighting, like ultraviolet light. And they did this by splicing in jellyfish DNA, and they were actually trying to come up with a way to make domestic cats resistant to feline AIDS. And they used a DNA marker to tell if their actual experiments were successful or not. So the glow in the dark traits of jellyfish were paired with the DNA that they really did want. And that DNA was DNA that would allow the cats to be resistant to feline AIDS. So instead of trying to infect the newer generation of cats with feline AIDS and seeing if it was resistant or not, they could just test for if the cat glowed in the dark. And if the cat glowed in the dark, then they also were resistant to feline AIDS. Now, that doesn't mean that if your cat at home glows in the dark, they're resistant to feline AIDS. And if they don't, then they are susceptible to feline AIDS. This is just an example. And it was done in a laboratory setting. Right? So exogenous DNA is DNA from another species that is put into a species in the hopes of improving it. And so there are three basic steps to this. One is to isolate the DNA from a donor animal. So you would take the DNA that you want. It doesn't have to be an animal, but a lot of times we're going to talk about animals. So donor species may be a better way to phrase that. So you're going to isolate that DNA. So you're going to use the stuff that we talked about last time in 5.1 where we talked about restriction enzymes that cut the DNA up, and then we're going to only grow the DNA segments that we want more of, right? So we're going to take that. Sorry, I hit my mouse and scrolled backwards. And we're going to insert that DNA into a vector, and a vector is going to allow us to infect a host with the DNA that we want them to have. And that's going to make something called recombinant DNA. Right? And then we infect the host with a recombinant DNA. And we're going to do that so that their own body replicates the new DNA in a way that gives their offspring the traits that we want. Right? So this is a very basic understanding of it. We don't need to go super in depth. In my class, we're also doing a GMO research project, and that's going to cover a lot of the same stuff as well. So this is in parallel to in-class work, as always. When we're talking about genetic engineering, we have a few ethical questions to think about. And as always, we are scientists first and foremost in my class, so we're not really caring if it's right or wrong. We're just saying, hey, this is a thing that happens. But ethics of this kind of thing do affect our lives as scientists and whether we should or should not do it, if we can secure funding for the research, and if it's something that will benefit mankind or not. So some ethical questions to bring up are, should we genetically modify crops to give them drought resistance or pest resistance? Because those could yield crops that are actually hurt by other specific bacteria or other specific pests or other specific viruses more. We have actually found a way to have bacteria produce some medicines, but that's humans affecting another living creature in a way that we've never been able to before. Is that a good thing? Is that a right thing to do? I don't know. I can't answer those questions, but it may be something that we need to discuss and talk about. Genetic engineering has led to the elimination of some diseases altogether. And that's a good thing because then people or species like plants and animals aren't affected by a sickness. So that's usually considered a good thing. But again, we are rewriting the DNA of a species. Is it mankind's role to do that? And as we get better and better at utilizing genetic engineering, specifically things like CRISPR, which we'll talk about later in class, we can change the way that fetuses develop by changing the DNA that they have in vitro. So we can have something that is termed a designer baby, where we can pick things like its eye color or hair color, or we can give it specific traits that we might want, say taller or shorter. Some of this stuff is things that we could do right now. Some of it is things that could be done in the next 5 to 10, 20 years, which is during your lifetime. If you're watching this video, it's probably during your lifetime. So it's something to start thinking about because it's something that will happen eventually. Right. So how is genetic engineering used by scientists? It's used by taking DNA from a donor species, infecting it putting it into a vector to make it a recombinant DNA, and then infecting a host with it so that its offspring will have the traits that we want. So this is something that's used. And like I said, we are going very over the top with this. We're not going super in depth. This is surface level. There's a lot more that we could talk about, but we don't have the time, and we are limited by the factors that bind us. So keep asking questions, keep learning. Have a good day.